fifth chapter, which is on the inheritance. So inheritance, some of the important checklists we're going to cover here is we're going to learn something called the concept of inheritance by the famous scientist called the Mendel, and basically how Mendel come out, which by his own experiments of the green peas. We're going to learn about inheritance. What is basically inheritance? We're going to learn certain terminology about gene, genetics, chromosome, and so on. First, we're going to start with the monohybrid cross. Okay, in this case, we have two plants. One is the one tall, and another plant is the shorter version. Okay, in this case, we're gonna see a certain few terminologies that is important because these terminologies have certain outcome in the certain in the cross. So an example, I'm giving you T. You can write any alphabeticals. For an easy example, tall, we just write T, and the T is here is the capital T, which represent dominant. Dominant means if they are present, they're always going to express themselves. If they are not there, they are not going to express. And if T is for short, so we're going to write lower case, the small letter T, and this represent the recessive allele. So they are basically not going to express themselves. If they are present, if there's only small t, and they're going to show it. So always dominant is the bigger boss kind of thing, and recessive is the smaller boss. So let's look at this chart over this side. P is the parents, and we have the gamete and first filial generation, and we have if the generation are being fertilize among themselves and what happened to the next second generation. So let's see the first parent. A parent which having the T and T, double T, it's going to be obviously tall. And another parent which have the smaller T, obviously going to be short. And since these both are having the same gamete, so we're just basically going to write one of the gamete as a representation. So we're going to write one T, which is the uppercase, big letter T, and the lowercase, the small letter T, representing the gametes. And when we fertilize them, they are basically going to form one capital T and small letter T, and all the plants going to be having a tall structure because T, which is the big T, is the dominant. So as I said, if the big T is present in the allele, and it's going to show it effect. So all the plants going to be tall in nature. Later part, if we fertilize among this generation, the first generation in between the siblings of the first generation, so we have the small t and big T and small t. Since they're having mixture of different combination, we're going to write all the gametes available. So we have big T, small t for this, and also the same big T and small t. And we're going to do all the crossing. After we do all the crossing, we can basically see we have three of the organism having a tall character because of the presence of the capital T here. This obviously 2, so obviously it's going to be tall. Since there's a big T here, it's dominant, so this is going to show the tall expression similar to this. But in case of the last one, since both are the short small capital T, small letter T, it's going to show a short characteristic. So overall, we can see at the second generation, it's going to form 3-to-1 ratio of the organism. So it's going to be 3 tall and 1 short. So these are an example of monohybrid cross. So in case of dihybrid cross, they're going to be more than one characteristics. Monohybrid is just single characteristic, either tall or short. In this case, you're going to have combination of the characteristic. So let's see, the characteristic are round and also yellow seed. Another one is wrinkle and green seed. In this sense, we have two characteristics, the shape of the seed, the color of the seed. So in this case, R, big R stands for the round, small r stands for the wrinkle, capital Y stands for yellow, letter small y stands for green. When the parents actually are being fertilized among themselves, we can see that the first generation basically going to form a round seed, which is yellow because the dominant R, the bigger letter, and capital Y, which is the yellow seed. So all the first generation gonna be all round and yellow seed plant. And later we're gonna fertilize among them. And if we want to do the cross in sense of by putting arrows is gonna be more difficult. So we need to remember that we can use something called a Punnett square. A Punnett square is basically writing all the gametes in a row and also in a column, and we're going to basically do a matching. So we're basically going to match the gamete at the column at this one side versus the row at one side. When we have the interpretation of the seeds, we basically can come up with the ratio. So in this sense, the generation going to have basically 9 round and yellow seeds, 3 round and green, similarly 3 wrinkle and yellow, and finally one plant will have the wrinkle and green seeds. So the ratio going to be 9, 3, 3, 1. So these are how we have two characteristic, and we call this system as dihybrid cross. So let's see again. Please always make sure that you do a Punnett square, because it's going to be difficult if you want to write in arrow form. So use a Punnett square for dihybrid cross. This is an example of a human right now. So we know that human have different blood grouping. So we call the A, B, O blood grouping. So human have either A, B, O, or AB. So what happened in different grouping, and what are something called antigen. Antigen is basically the marker that present on the red blood cells. Antibodies are something that present in the blood serum, the blood plasma. If you remember our component of our cells, we have something called plasma. And whether we can donate or whether we can receive, in which group we can donate, which group can receive these groups. So let's look at the blood group A. Since we say the person having blood group A because they have an antigen A. Since they have antigen A, they can't have antibody A. They need to have opposite of A, which is the anti-B. We either have anti-A, anti-B, or no, not at all. We don't have any other anti. So a person who have blood group A will have antigen A, and the antibody is reverse of it is called anti-B. Since the person is since to have the antigen A, the person can only donate someone who have the A blood group or AB, because they also have A. Since in this case, they can receive person A who lost blood can only receive blood from A, their own group, or O, which is said to be universal donor. In case of B, similar, we have antigen B. Opposite of this, the antibody going to be anti-A, and the person can donate to their own blood group, which is B or AB, and they can only receive from their own kind, which is B, or the O, which is the universal donor. In case of AB, the person is said to have both antigen, A and B, that's why they are AB. Since they have both antigen, the blood should not have antibodies against this. If not, it's going to attack your own blood and resulting in death. So too bad this person can only donate to AB, since they have because the person will have antibodies against this. So they can donate to AB people. But the good part is, since they do not have antibodies, they can receive from all the blood group A, B, AB, and O. All of them can give the blood to them. So AB is quite lucky. In case of O, they are not having A, they are not going to have B antigen, so none of the antigen present. And since they do not have antigen, they will have antibody, which is anti-A and anti-B. As I said, O is universal donor. They can donate to all the people, AB, AB, and O. And too bad, since they do not have, since they have the antibodies, they can only receive the person from O. So it's a bit difficult to identify at this level, but if you see the next one, you can further understand in more of the genetic level. So a person who is said to be blood group A here, okay, blood group A, the genotype is basically telling the gene responsible for it. So we're going to write IAIA, which stands for the blood group A. If they react with anti-serum A, they're going to form something called agglutination. So there will be clumping happening, and this is a positive reaction because the person is actually reacting towards the anti-A. If the blood group A will not form any agglutination, so they're negative, so the negative result going to look like this, which is not an agglutination. Person who have a blood group A also can appear to be A and O, because one of it are still dominant, so they're going to be still showing a blood group A. So in this case, the agglutination is going to be the same. Blood group B can have two genotype, either double B or 1B and 1O. So in this case, the anti-B serum going to be positive and forming an agglutination similar like this. Okay, for blood group AB, obviously one going to be A, one going to be B. There's not going to be any other combination. When we react with anti-serum A and anti-serum B, obviously both going to form the agglutination because they have reaction towards anti-A and anti-B. So these are we can see in real blood group testing. These are the testing that happening in the laboratory if person want to test their blood group. And finally, for blood group O, they're going to have only OO genotype. And since they do not have any antigen reacting toward it, so even though you put anti-A and anti-B, they're not going to be any reaction. So they're going to be a negative reaction take place. So these are overall of how a person blood group can be determined when a person wanted to know their blood grouping. So this is about AB blood grouping. This is another important part of the chapter of inheritance, because basically we're going to deal with something called DNA. The long name of DNA is deoxyribonucleic acid. So basically stands for DNA. So we have know that cells is the basic structure of the living things. We remember from 4 chapter 2 cells, we have animal cells and plant cells. So all the cells will have the nucleus, which is located here in the middle, which is a cell nucleus. If we able to zoom by using microscope, by using a high electron microscope, not the normal microscope, we basically can see something called a chromosome. Chromosome is like pad, like a Y shape here. And further, if we able to zoom into this chromosome, we can see something called gene. And these gene actually are coiled in a strand called the DNA. And in the DNA further, we have this colored here, are all called the bases. So we have certain bases, which is gonna be forming a bond between them. And because of forming a bond between them, they're gonna form something coil structure called the DNA strand. All the coil structure gonna actually become thickened to form a chromosome. And this chromosome basically located in the nucleus. Okay, so if we able to zoom the structure of the DNA in a more diagrammatic way, we can see the structure are basically composed of nucleotide. So if it's difficult to see here, this is the structure of a nucleotide composed of a phosphate group, the blue color, the sugar, which is the red color, and finally we have the nitrogenous bases, the yellow color. So each of these structure are forming a bond in between this nitrogenous base, are forming a pair. When they form a bond, which is called the hydrogen bond, they are basically forming a structure that look like coiled, known as twisted, and they form something called double helix. So this is how a double helix being coiled, forming a structure of DNA. So we know that nucleus have the chromosome. Chromosome will actually basically formed by the coiled of the DNA strand. And DNA strand is basically made up of something called nucleotide, which have phosphate, sugar, and nitrogenous bases. Combining each nucleotide, we form a twisted structure, which is called the double helix. So these are the basic of DNA. We have the phosphate, stand for P, and we have the S, which stand for sugar. And here we have some letters, which is A, T, C, G, and all of these are called the bases. So the name of A is referring to the name of the bases, which is adenine. T stand for thymine, C stand for cytosine, and G stand for guanine. Always remember, adenine will always combine with thymine. The rules are set saying that A combined with T, C combined with G. That's why you can see that A, T, there's no other combination. Similarly, C combined with G, C, G, CG. The rules are set, so please always remember A combined with fused with T, C combined with G. Okay, later we're gonna see these are some of the application of learning genetics, chromosomes, gene, and certain things here. At the part of this part, we're gonna learn something called the genetic engineering. Basically, these are being used widely to produce modified organisms. Like in case of we wanted to produce human insulin, like for example, basically human insulin are produced from the porcine. Um, and this is how the steps are going to take place. So let's appreciate the steps here. So we have the human cells which able to produce insulin, and in the middle are the DNA structure. And to produce this combination, we also need a bacteria that have something called plasmid. You can see in the blue structure here. So basically what we're going to do is we need to take out the gene responsible for insulin, okay, the red color here. And we're going to take out the plasmid from the bacteria, which is the blue. We're going to, since it's in a round shape, we need to cut so that this red can combine into this blue structure. So we're going to cut this, make it open, so that the red color structure can combine with the blue structure and forming a round shape of something called a recombinant DNA. So take the DNA part, which is the DNA of an insulin, take out from the cell. We're going to take out the plasmid from a bacteria, cut it, combine it, and now we are forming a recombinant. Now we're going to put this recombinant into a bacteria which is able to multiply, able to actually produce more copies. So when the bacteria grow, the recombinant plasmid also grow. So at this one stage, we can see there's a lot of cells producing insulin. And later we can purify these. And basically at the last part, we are producing a bacteria which can produce more insulin, and purify this. We can actually use this insulin to treat diabetic patient. So these are one of the way where genetic engineering is taking place. We need a part of DNA, we need a plasmid from bacteria, combine it, put it into bacteria. As the bacteria grow, it produces also these insulin with them. And finally, this insulin can be used for diabetes patient. So let's recap about inheritance here. So we saw how Mendel law is important in sense of monohybrid, and monohybrid will produce the second generation uh, with a phenotypic of 3:1. Whereby in dihybrid we have two characteristics, and the phenotypic is always 9:3:1. So these are how Mendel come up with a law of segregation, and also law of independent assortment. They can assort among each in different combination. We also saw about gene and chromosome. We appreciate the structure of DNA, which is the double helix, and we can see how these structure are important in the nucleus for genetic control. We also see some of the ABO blood grouping. We didn't see much of these sex chromosome, but we'll see in the question later. And we saw how this knowledge is important to be applied in a lot of genetic field. For example, we saw how we can produce genetic engineering with genetic modified food. Stem cell research is important right now because stem cell is basically can divide into many other cells and can be used to treat maybe cancer and also some infection. Gene therapies is also widely being used. It's coming up slowly. Human genome project is basically where they actually sequence or get the these sequence of a DNA, so a person have the full genome can be used to treat disease also. So let's see the question about DNA structure here. So we have two structure here. One is in easy way, we can see they are coiled, double helix, and we have some base structure. So by now you already know this P is belonging to something called the DNA. On the other hand, when this being coiled, forming a thickened structure and like a Y shape here, it's obviously easy to identify. This is a chromosome. So this is how DNA and this is how a chromosome look like. So the next question is to see the relationship between P and Q, how basically P later become a Q. So Q is made up of all this P, become all the coiled, and they're going to be tightly made into a structure. This is going to be into a tight structure. And you can see each of these strands are DNA. They are tied by a protein. So this is how this P can be converted into Q chromosome. Okay. The further question is how we're going to see the part make up the structure P. So I already said P is basically made up of nucleotide. It consists of sugar. Um, and we have also phosphate group, and the one seeing here colored part is all the nitrogenous bases, and the function is easy. You can say it basically determine the genetic characteristic. If a person is appear to be like a mom or like a father, is basically the mom and father are giving the instruction for the baby to be appear to be these similar. If the parents have certain diseases that they can be transferred to the baby will have certain disease which is the genetic inherited. This question is basically about homologous chromosome because it's pair. This is why it's called homologous chromosome. If it's not pair, it's called just a chromosome. So we have set 1 and set 2 here. So the question basically asks you to identify what is this K and L. K is bigger and L is smaller. So in set, okay, always remember that in human, X chromosome is bigger. So that's why it's a bigger in shape. So K is actually X chromosome. By Y chromosome is smaller. So in this sense, this is how X and Y chromosome homologous chromosome structure going to look like. In set 1, this is an X, this is also X, so it's quite different. The next question is to identify the gender of a person. If this is X and this is also X chromosome, obviously it's a female. Female have X chromosome, like male have XY chromosome. So that's how we differentiate. So sex chromosome for female is XX, for male is XY. This is more of dihybrid cross. So the question already give you the clue. Big R stands for the dominant allele, and which is round, and this is yellow. So we have to basically come up with the phenotype, the characteristic that you can see. Plant 1 have double big R, which is double, and plant Y, plant 2 have small r and big Y. So this stands for round, and small y stands for green. So please use these to interpret the characteristic similar to plant 2. We have seen the wrinkle and also a yellow seeds. And the later question is to complete this. So please take, we know that both are the same in sense of this R double Y. So just please take one of it to write the gamete out. If it's same, please write only one. If it's different, then you have to list all the combination. State the phenotype of the first generation. So if this first generation, so basically ask you to just interpret this. So it's obviously round because of the capital R, and also the capital Y stands for the yellow. So in this case, it's going to be round and yellow, dominantly expressed. So this is a question about how process of mutation is taking place. So in this case, we have A, B, C, D, E, F, G, a chromosome part. And later this chromosome part, we just only have A, B, C, D. E, F, G are missing. So in this case, something have taken place. A deletion is basically taking place. All the E, F, G have been deleted. The next question is to, first you need to identify the karyotype. All these arrangements are called the karyotype. So the question is asking about determining the gender of this person. Since we have X and we have Y, it's easy. Please look at chromosome 23 and we know that X and Y belong to the male person. And this person is said to have some kind of genetic problem. And by looking, we have all the chromosomes are in double, in homologous, except at the number chromosome 21, there seems to be a triple arrangement. And this is not normal in sense of normal person. So if they have extra chromosome, so if they have extra, this person is says to be Down syndrome because there's 3 copies. This only should be only 2 copies. This question is about how we have 2 types of mice. One is a brown fur, another one is having a white fur. When these two are being actually mated, we can see all the babies are producing only a brown fur uh, babies. So why the offspring, the babies are only having a fur which is brown? Because in this case, the brown fur is dominant. Because all the offspring having brown, so we know that the brown is dominant. So all will carry the dominant characteristic, the allele, and they will also have white, but the white will not be shown because white is recessive. So all offspring will have being in case having the brown and also white, and the brown is more prominent.